So I have a presentation myself that is a little more sort of data driven, and just sort of examples of how we can use data sources from across the fields we're interested in, and how useful it can be when we get data sets together in the same space to look at them. You know, we can obviously think of things that we looked at in Nicolo's data today that would make sense here. So if you have 6 donors in a single organoid and you have a description of a neurogenic trajectory, can you look at the difference in the 6 neurogenic trajectories taken by the 6 individual donors, for instance? So you can think of that in a single experiment, but we can also expand our thinking to incorporate wider data sets, mouse, monkey, and human development, particular times in development, multiple donors, and things. So very quickly, if you're thinking about these problems, you want to get lots of data sets. And so the past years of work with me and many colleagues that some of you know on this call, Ron Mackay, Sulki Kim, Sung Mei Seo, we've been collecting data sets and trying to understand them together. And so I'll show you everything I'm going to show you. I'll put links in the Slack channel and emails to the list, so you don't have to worry too much about scrambling to find out what the links are. But everything I show you is totally functional and public, and you can explore them just as we do in this session afterwards. And I'm realizing it's close to the hour. I'll probably talk through the end of the hour. Stay if you can, and you can watch the video later if you can't. So this is a web page in Nemo Analytics that is focused on development a little bit earlier than we have been talking about today, right back at the gastrulation stages, and even earlier when we see. But the Nemo Analytics is a web resource that I have been developing along with colleagues at the University of Maryland, also here in Baltimore on the East Coast. And so each box you see here is a data set from a public data set that we downloaded and formatted so that we could plot in sort of a biologically informative way. This first one is from Peng et al., which is a study of microdissected embryos. So these are 3 different mouse egg cylinders that were dissected 11 slices in their proximal to distal range, and then each of those sections was quartered. So this is sort of a coarse resolution physical map of the mouse egg cylinder, and then each one of those samples was individually RNA sequenced. So this is simply a spatial map of what they are. And once you go to this link, you can just type in a gene. This is brachyury, which I'm sure many of you know from gastrulation fame and a lot of mesendodermal lineage definition functions. So I want to start this as a discussion. So if you want to see a particular gene or you don't understand what particular data set we're looking at here, jump right in and slow me down. But I want to show 2 things. One is the utility of just getting these data sets next to one another and in the same space to look at. And then the second one is, okay, once we have them all together, without building an entire coding environment to harness that data collection, which we're also doing, what small number of tools can we give a cell biologist that doesn't know how to code? What tools can we give them in this graphical user interface that can allow them to do more biological searching beyond individual genes? So we'll look at some individual genes, get familiar with some of these data sets, and then I'll show you 1 or 2 more things we can do in Nemo Analytics that really expand what we can look at. So from going from individual genes to then gene lists and complex gene trajectories that we get out of dimension reduction, so that we would end up not only looking at a single gene across data sets, but an entire trajectory or transcriptional module that was discovered or defined somewhere else. So before we get into all that craziness, let's just get used to some of these data sets, so later when we're looking at pictures, we know what those pictures are and what they mean developmentally. So here in this first study of 3 embryos, they're E7.5, and everything, by the way, in Nemo Analytics, anytime you see a data set, if you click in this upper right triangle here, You can go directly to the publication in PubMed. If the data is in geo, there's a link here, and there's also an info button that has the name and date of the study. If they have their own website where you can also see the data, we always link to it here, so they do. They have an iTranscriptome data set, so this is a website dedicated only to their particular data set. So if you're ever interested in any one data set, you can always go to this side button here and get more details. Okay, so here we see the gastrulating side of the embryo, the side of the embryo that is listening to the BMP signals coming from the placenta, or the extra embryonic tissue here. And that's the posterior side. That's where brachyuria is turning on. And on the other side, that will eventually become the neurectoderm, nothing is happening. It's being inhibited by a whole system of different tissues, including the anterior visceral endoderm on that side, that basically make these cells wait to do anything until they can become neurectoderm. So this is very spatially defined. The next panels here are 6-hour increments in a study from P1 Sala et al. And we can show you that one. This is from Bertie Gutkin's lab. This is one of the most sort of famous early developmental studies. Many people recognize this image. It's E6.5 to 8.5, as I said, and they did 6-hour increments, so a really nice high resolution time frame here, or high or low, depending on what you want to look at. But they defined 37 different lineages here, labeled as clusters on this two-dimensional map of the high-dimensional data. And what we've done in NEMO is actually taken each of those 6-hour time points and shown them separately here. So this is the first one at 6.5, where you can see the epiblast, and this is extraembryonic ectoderm and endoderm here, with no other lineages yet emerging. But you can see already at the beginning of gastrulation here at E6.5, brachyuria is turning on in the cells that are about to gastrulate, and you can kind of see them send themselves off in this north direction, which is mesoderm, and the south direction, which is endoderm, as gastrulation begins. And so it's really kind of a flip book of time in the mouse in these 2 days that are very important in setting up the entire body plan. So again, just getting you familiar with a couple of these data sets before we go on. I'm going to skip a couple here. This is a very similar map to the one we just looked at, but this is in I think it's cynomolgus monkey, the Macaca fascicularis. And here again, you can see streaming out of the epiblast, the gastrulating cells of the macaque here. These are some earlier times from the Borowiak group, quite earlier. If you look at some of these, they'll go all the way back to the 4 and 8 cell stage here at the beginning, and zygote. And they really do this is not single cell, but it is high resolution microdissection followed by RNA sequencing. And across a couple different studies, they have mouse, marmoset, and human data with these two bigger, more sort of in-depth studies in the marmoset themselves. So these genes often play an entire game in the early embryo before they get to the role that we might know them best for in gastrulation and so on. Brachyuria waits to come on, but another one that we recently looked at was Mixel-1 that has an entire something happening early in 2 and 4 cell stage. It goes off and then it comes back during gastrulation. This is a Petropolis study from 2016, human cells from embryonic day 3 to 7 across several embryos. And there's tons of these different ones. And again, feel free to come here just to learn about these. Each one of these is a whole study that you can go run to. This is from Jay Shenjuri's lab out west. They've done a lot of early mouse and early in vitro modeling. This is from a Chinese group, Chen et al., a year or two ago, doing stereo-seq in slices. So you can see actually for brachyuria, it's a little late to be looking at the embryo here at E9.5, where gastrulation is wrapped up. So the data is very sparse, but as you'll see, once we aggregate genes that are functionally related, you can get some exquisitely detailed views of the sliced embryo that aren't simply the staining with a single antibody or the labeling with a particular RNA to see where it's expressed, but actually multi-omic data at a microscopic tissue-defined level. So that it's becoming more and more mind-blowing, the kind of data that's out there. And so I'll show you some projections that are a little more complicated than individual genes that really harness the energy of these spatial data sets in a moment. So this is the individual gene world. One other study I want to bring to your attention, whose data we're still bringing in, is this one. It's Zhang et al. Just in the last year or two. Also, Oscar Marin is involved with collaborators from Beijing here. But they had some of the earliest human data that's out there, and also some of the best time course of this early data. So they have 3 weeks to 12 weeks of development in the human. And in that first time point, the 3 weeks, this is their single cell data from that early time point. And in addition to the data itself, they did a very simple thing here, which is do some differential expression and get a gene list that's basically markers for each one of these lineages. And they've marked 19 lineages on here. And I'm going to use this as an example of how we can harness a component of one data set, which is lists of lineage-specific genes at 3 weeks of development in the human, to explore all those data sets that we have on NEMO. So the next tab I have open here in NEMO is the exact same collection of data sets we were just looking at. You'll recognize these as the same 3 egg cylinders from the mouse that we were looking at before. But instead of brachyury now, we are looking at the aggregate expression of all the genes that Zhang et al. Annotated, in this case, to cardiac myocyte. So these on the left here are simply the gene lists that were in their supplemental table 2. They had a supplemental table, I downloaded it, and then uploaded it to NEMO, and then told NEMO I would like to project or look at the strength of these different gene lists in other data sets. So this is the aggregate expression of all of the genes that were labeled as markers of cardiac myocyte in the Zhang et al. Study, now shown in the other studies. So in the egg cylinder, there's not a whole lot of order to this cardiac myocyte signal except right here. This is something we've seen before. We think it's actually a piece of the anterior visceral endoderm that got caught in a particular slice. It often has very extreme expression of things. But that's kind of a little detail we shouldn't get into, I don't think, right now. But to kind of show you how well this works across these systems, this list of cardiac myocyte genes, if we look at the later Pijuan-Sala data time points, this quite surgically lights up the lineage here that they themselves define in their study as the cardiac lineage. And you can really see from beginning to end when this is the first time point, maybe at 8 days. Let me move here. Even at, looks like 7 3/4, you can see that cardiac signal emerging. It's also common to back here where these, and I can, I think, flip back to it, are actually representing, so this is the heart here in purple. Back here they have what they're calling mesenchyme, allantois, or up here 26, what is endothelium. Now, coming back to NEMO, if we look at this, where the actual cardiac lineage emerges, we have a really high level, about 250 units. And units become a little crazy when you do projections. So they're really only, we don't want to go comparing the units we're seeing in this Pijuan-Sala study to, say, this egg cylinder study. But inside the Pijuan-Sala study, here we see the cardiac lineage emerging, and we're all the way up here at 250 units. This signal also was emerging back here in that mesenchyme zone, but it's at a much lower level. So you kind of have to watch out with your oranges and your purples and your time scale, excuse me, your color scales in which study you're looking at. But clearly it's the heart in both systems that are really showing this. And it works across the board. Those 19 lineages really paint quite a precise picture across the mouse development we're looking at here, despite the fact that they're from the human development in the other study. And of course, we would expect this. But again, really what this is about is bringing together the totally insane resources that we currently have available. Now, available is a kind of a relative term here, because if you actually wanted to get all this data and ask these questions, and you didn't have them all assembled, it would be, you know, weeks to months to getting all the data together and then start to ask questions. So we're really trying to provide an environment where all this has been done, where the data is ready to look at. Jump right in, Nicolo. What's your question?
Thanks, Carlo. I think this
Yeah,
is a great resources. I had maybe a technical question, but we were working on that on different projects. So my question was, how do you compute the aggregate score? Because the reason why I'm asking is that, so for example, in SCAMPy, there is a formula that basically average out the expression of the genes that you have in your list and subtract it to a random selection of genes of the same length to have a sort of normalizing factor. However, so imagine that you now have two cardiac myocyte signatures that for some reason are not, you would be interested in a slight difference among the two. What we have not still realized properly is how to tackle the length of the list that you use, because since one gene highly expressed, for example, can have an impact on the average, I was wondering if you were
yeah. So there's a couple ways we do it. So the concern you're mentioning, which is, say there's 10 genes in this cardiac myocyte signature, and maybe one of them is expressed 100 times the others. So really all you would be looking at is that one gene, right? So there's a couple different ways of doing things. This here, where it says select algorithm, actually determines the calculation that goes into taking this gene list and taking this data set and then ending up with a display. So there's a couple, if you actually check these different boxes, that math is run in different ways. So currently, we don't actually have something that just popped into my head that would address your concern, which is, say, some kind of robust mean, or even a row scaled mean, where every gene contributes an equal part, which would require a row normalization ahead of time. What we do have is so because we were thinking of using this not only on binary gene lists, but also on gene lists that have particular weights generated with things like PCA and NMF, here I'm using a binary gene list, but I'm using the math of PCA. So what it does, it assumes that all the genes in your cardiac myocyte list have a weight of 1. And then the way PCA math works is that you would take the weight of a gene in PCA, multiply it by the expression in the sample, and then sum all of those for all the genes. So if you have 10 genes that are in your cardiac myocyte, you would this is simply a sum of their expression. But that suffers from the same potential problem that you mentioned, is that some of them will be speaking louder than others, right? So we'd like to add a normalization.
Already very interesting to have the weights on the, yes, thanks.
Yeah, but in this case, it's all sort of agnostically averaged. So I think a great idea would be an added little checkbox here that says, okay, give me the average, but make sure every gene speaks with an equal voice. You have a flip side problem there, though, that if you have 10 genes and one is really high, maybe only that one is speaking. On the other side, if you have one that's very low and perhaps only represents noise, if you normalize them all, then that noisy gene will be speaking just as loud as the others. So there's ways to temper this, but I think it's good to have options. The other ways we have here are really just for if you have a gene list that came from PCA or in this case, we have two different ways of using NMF weights that you can project. The other thing we have that's very useful that doesn't appear on this page exactly, but is relevant to the kind of binary gene list we're looking at here with the cardiac myocytes is it's called just a binary gene count. So if you have a list of 20 cardiac myocyte genes in your single cell data, instead of trying to figure out what expression level they have, for each cell, it simply sums the number of those 20 genes that is non-zero. So maybe 10 of them are expressed at some level and the other 10 are zero. And then each cell is then colored by how many of those genes appear at all in your cell. Again, that has pluses and minuses. That stops the problem of one gene being very high, but of course there's noise at the low end. But again, having options in how we project these gene sets into the new data sets is kind of the solution there. But it's a very big concern, because if you're not thinking about how that math is done, the picture you're looking at could be interpreted in very, very different ways. Okay. So does anybody have any particular lineage they might be interested in, or a gene? Or should I things you'd like to go at from here? I think we can maybe go till half past. We have some time to play if we want. May says, let's look at the notochord. Okay, now, so here I'll be selecting notochord from the list. So now this is just another set of genes that was in the supplemental table 2 of that Zhang et al. Paper. And so now it's pulling up all the same data sets, but instead of looking at the sum of the genes that were annotated to cardiac myocyte, we're looking at the notochord genes. This is surprising to me, actually. I would not have guessed that we could already see this signature in the E7 data, where I'm guessing the notochord, the involution is just beginning here, and it's right about where it should be there at the most anterior portion of that gastrulating side. And then we can look at this same signal now across other data sets. In the P1 Sala et al., it's highest here in the extraembryonic endoderm. But again, we have to watch this. We're at 50, 50, 70 units. As we move to later ages, the units are much higher, and the notochord itself, I know this data set quite well, so that's what this little branch is clearly lighting up in the mouse, as we would expect. The human genes expressed in the notochord we would expect to be precisely the same ones as in the mouse, but again, just showing that this kind of system works here. And here in the downsampled entire P1 solid data set, you can see the notochord really lights up here. It is interesting, we can see it already right as it's beginning to form and before neural induction is happening here in the E7 embryo as well. So this is some data from the Lamano paper from Sten Linnerson's lab that had this was actually published as an atlas of the brain, but at early times, they sampled either the entire embryo or the entire head of the animal. So here, let's see, this is E7. This might be the notochord right here. We'd have to go in depth a little bit more. And in fact, it wasn't even annotated in the paper. As I said, they were really interested in the brain in that paper. But at E7, this is probably the same signal as we're seeing right up here, because this is another E7 mouse, just very different structured data, but beginning right there, a little earlier than we might expect. Here is the macaque data. This requires a little bit of color matching here. So whatever color this little subpopulation is, we would map here. I don't know if they have notochord NC. Is that notochord? That's not what this is, though. Where's that dark blue? If anybody sees what might be, they have the node there. That looks like the node to me.
What they call node.
Yes.
And,
okay.
and everything you're talking about is called node, actually. All these early, at the top of this, the early distribution and the mouse egg cylinder, right. Everything else, yeah.
before it actually involutes and goes under the presumptive neuroectoderm as the
But
notochord.
before it becomes restricted to the notochord, right? Yeah. Or not really. This is the whole ancient. Yeah. This is Feynman we're talking about now.
Didn't they just have a big anniversary of the Spemann-Mangold organizer paper? 100, is it 100 years or no?
Yeah, and there's an interesting meeting actually this year, I think, in Europe, or on self-organization, which is of interest to the group on this call, I expect.
Yeah, I think it is 100 years, right? 1924. I think that's right. Very cool. Thanks for that, Ron. So it is noted. Some of the colors here become a little hard to distinguish. We haven't broken this macaque data out into the individual days and time points as we have with the mouse, but I'd really like to do that. They have Carnegie stage 11 to 18, or excuse me, 8 to 11. That would be nice to see broken up. And this is a problem. I have not input the orthologs for the marmoset, so we don't have those here yet. So all in the background, of course, is not only projecting the signals, but mapping the genes across these different species. This is interesting now. So these are human data sets quite earlier than we would expect a notochord, but the transcriptional program is arising. And this is interesting in the Tyser data. This is a Carnegie stage 7 human embryo. The Petropolis is even earlier. This is day 3 to 7. It looks like in day 7, looks like epiblast, perhaps even primitive endoderm is starting to really show where this will be emerging later as the notochord. Okay. So I'm wondering now which other steps. So actually, maybe what we should do now so we've been focusing on in vivo development, particularly here. In the last, say, 10 or 15 minutes, we can move to another set of data sets. So all the data sets in these boxes are now different, and I'll explain what they are, but our lists of genes on here on the right are the same. These are still the 19 lineages from the 3-week human embryo from Zheng et al. And here, instead of having a collection of in vivo data sets, we have a collection of in vitro data sets. This is from our manuscript, who some of you may have heard a month or two ago talk in the same series, Sol-Ki Kim, on variation in the earliest steps of both pluripotency and the differentiation of pluripotent cells, where we studied 2 sets of 6 lines to look at their differences and how they develop and why, and then broadened that to look at collections of hundreds of IPS lines that had been already sequenced as part of consortia projects. So again, we're looking at the same gene lists over here, whether it's cardiac myocyte or if we look down at the notochord, but in this case, different data sets. So here in Sol-Ki's data, we have 2, 4, and 6 days within pluripotency, and the colors are 6 different lines. This is 2, 4, and 6 days of dual SMAD inhibition. So on our way to organoids or any other neural differentiation we might be thinking about. And then this last 2, 4, and 6 is BMP induction or mesendodermal differentiation. So BMP4 application, where we see a big jump in the signal here, where the pluripotency and neuroectoderm differentiation are going nowhere. But here, all of the lines respond by increasing markers of the cardiac myocytes. Even though there are no cardiac myocytes in this system yet, the transcriptional programs initiated immediately by BMP when exposed or when put on these pluripotent stem cells, these transcriptional programs are already revving up. Again, even though we don't have specific cardiac cells yet. This is an experiment where Sol-Ki used SOX21 knockout cells from one of those lines. So this shows that the induction of BMP is usually shut down. So this is day 2 treatment. If you put BMP on cells 2 days after you plate them, most of their ability to react is gone. That's the wild type cell down there in black. But cells lacking SOX21, which is a key neuroectodermal regulator, are still responsive to BMP. This being the cardiac myocyte response to BMP, but that's part of a much larger transcriptional program that's happening here. So we can see that in the knockout, which is interesting. And so now I'm just going to show some other, again, this is the same cardiac myocyte signature. This is another data set worth pausing on. So when I said we looked at hundreds of lines, the data came from this study. This is Ihor Lemishka, a study from 2017, the Next Gen Consortium. So this is 101 donors, and they have 317 lines from those donors. So this is simply a PCA plot of their data. So each dot is a line. So there's 2 or 3 dots that come from each donor in this. And clearly the cardiac myocyte transcriptional program is active in many of the cells in this pluripotency. There's no differentiation in this data. But clearly PC1 is defining an axis that is nearly perfectly parallel to the cardiac myocyte signature, with high PC1 values having a lot of this and low PC1 values not having a lot. So this is showing, one of the things we put forward in the paper that all this data is from is that the variation in human pluripotent stem cells within pluripotency actually contains the dimensions along which the people's lines differ are the same as the lineage map of early development. So, this shows that the PC1 of 300 lines in pluripotency aligns with the strength of their cardiac myocyte signature. Again, way before any cardiac myocytes exist. And so that cardiac myocyte signature is probably a proxy for the very large BMP-driven mesendodermal signature that we know to be very high at this end of things. And this end turns out to be neuroectoderm. So actually, let's see if we can show this here. Let's look at neural tube 1. They have 3 different neural tube signatures from that 3-week mouse human embryo. And here we see, again, the in vivo signature of the neural tube mapped onto a PC1 and 2 plot of 300 human pluripotent stem cell lines before they differentiate. So now clearly the lines that had a high idling of the cardiac signature have a low idling of the neural signature, and vice versa. The cells over here that had very little of that cardiac myocyte signature have much more of the neural tube signature. So there's this sort of whirling balance of the lineages that will emerge in only days or weeks from these cells in normal development. But already in pluripotency, they're brewing and they're whirling. And what's important to know is that not only us, but many others have shown that this is a stable trait of cells from particular donors. And I'll show this by showing a figure from our manuscript where this data figures, where this is actually another representation of that same next-gen data, the 317 lines from 101 donors. They're ordered by PC1. Their value, they're ordered by PC1 from that donor. So if there's 3 lines from a donor and they're all high, they're out here. If there's 3 lines from the donor and they're low in PC1, they're here. But what you'll notice here, and actually let me zoom into this, and this can be sort of the last point that we go into. So what you can notice is that there's very little vertical variance. And there's some cases we actually highlight one where this is, any vertical line is one donor. So this is a donor from, or a line from the same donor as these. So this is a sort of rare departure of lines from the same donor, which means that this trait, or this quality, is actually a stable trait of these lines. And we've seen it in our 2 sets of 6 lines. We've seen it in these collections of 100 lines. So these 2 things put together mean that pluripotency as we know it in a cell line or in collections of thousands of cell lines is not a singular state, but sort of a dynamic state where all of the lineages that will come are already whirling around, and they're whirling around in a particular balance in any particular donor. So you might be, now this doesn't mean that some person is going to make a heart and the next person won't make a heart. We know, of course, that canalization during the development and in the uterine environment ends up building a human with all of these pieces. So we don't really know what it means to have more of a cardiac signature early on. We do know, though, that some of these things can be teased out when you look deeper into the transcriptome in pluripotency of these different lines. For instance, in our manuscript, we note that particular retinoic acid response genes, including GBX2, are more highly expressed in some lines than others. And when you put those different lines through neurectoderm differentiation, you get very different numbers of either dorsal and ventral neurons, but in relation to GBX2 in anterior posterior regionally defined neuronal populations, where GBX2 and OTX2 play an adversarial game around the mid-hindbrain border. And so cell lines that express more OTX2 on average during pluripotency actually have, when you put them through dural SMAT inhibition, produce more anterior neuronal fates. And lines that have more GBX2 expression end up making more neural fates characteristic of posterior regions like motor neurons of the spinal cord, or even midbrain and hindbrain neuronal fates as defined by different Hox gene markers. So again, very happy to surf any particular questions that others may have here, but I wanted just to give you a little bit of a flavor of what we can begin to see when we get all these data in one place. And maybe as a last demonstration of this, I'll go back to the cardiac myocytes and look at brachyuria alone. It was very sparse, and most individual genes are quite sparse. Oops, this is the wrong, my bad. That's the wrong profile. In the spatial data, it was so sparse. If we use projection of a large number of genes to get a signal, things get a little more clear. So this is that same spatial data we were looking at, the E9.5 mouse embryo, where each one of these dots is a spatially defined unit where sequencing was done. This is the cardiac myocyte signature, the differentially expressed genes from the 3-week-old embryo from the Zengadal study, and it's projected right into the entire spatial data set here. And as you'll see, what is defined here as the heart is absolutely glowing for us. So there's no question that are quite surgical in lighting up the cardiac cells in the mouse embryo, even though they were generated in a study in a different time, in a different place from human tissue. And again, just trying to show you the use of here. If we get the data together and all of a sudden we are able to bring in signatures from different studies, we can really ask a lot of questions. Yes, so May just put it in the chat to let you guys know that if you're into using Nemo, you can not only look at the data that's there and the gene lists we've uploaded, you can upload your own data set to Nemo and keep it private while you explore it yourself. You can add other data sets that are public to your own in a profile, so you can look at your own data as you're writing a paper or exploring it beforehand. You can also upload gene lists, simple list of your 5 favorite genes, or more complicated things like PCA with gene weights. And also then you can use it to explore, but when you're ready to publish, you can use the site at either permanent links to your permanent data sets as you set it up, so you can use it as a accompaniment to a publication so people can explore your data. We would recommend it as in addition to a repository, but it's much more usable here immediately for biologists, especially without coding expertise. Yes, Nicola, or Nicolo, jump in.
Thanks again, Carlo. I think the discussion is super interesting, and I think we will upload also our dataset as soon as we have permission. So
Oh, wonderful.
Nemo is wonderful. I had one question on the previous results that you were discussing. So different colleagues in the lab have shown in previous work that at the IPS level, you can capture transcriptional features that resemble the disease of the patient in which they were reprogrammed. So this was the case, for example, for Guillain-Barré syndrome and 7-DOOP that are two neurodevelopmental disorder, but also recently on a different disorder in which you see strikingly that already at the IPS level, the transcriptomic differences between in several individuals carrying mutations for these disorders and control individuals were altered in domains that were a feature of the clinical manifestation of the disease. So I was wondering if you observed something like that in the data sets that you are characterizing, meaning that you can in a way correlate the transcriptional alterations between IPS with some clinical covariate of these cohorts?
So we haven't gone, we haven't covered that much distance all the way from IPSL to clinical, but we have covered the distance I mentioned just a bit, looking at iPSC transcriptional traits and differentiation traits. So in fact, I think very quickly I could show you one other piece of evidence here. So, lucky 13. Okay, so these are the same stem cell data sets I showed you a minute ago. Now, instead of looking at the lineage defining gene list, we're looking at 22 patterns that we define simply by decomposing this data set. So if you decompose this set of 6 lines in 3 conditions and across 3 times, 2, 4, and 6 days, you get lots of patterns. For instance, we get one that defines neuroectodermal differentiation here. You can see it going up here in the neuroectodermal differentiation with nowhere else. But something else that was super interesting to us, and this is getting more to your, I think your thinking here, Nicolo. All these purple dots are the same line, H9, maybe one of the most famous ES lines in scientific history. But what's interesting is that in a set of 22 patterns across these 6 lines that were all differentiating, we got lots of patterns that were about BMP mesodermal differentiation, a couple that were about neuroectodermal differentiation, but then a couple, actually exactly 6, that look like this, that's a cell line. It's not about what time it is, it's not about what lineage, it's something that is particular to this line. Now, it's important to remind you here that this is an NMF, so there's not many genes that look like this. We know there's not enough genes in the genome for each of us to select the genes we will use to build our bodies. We all use the same ones. So the signatures from an NMF are components that are combined to generate the expression of individual genes. So if there's a gene that comes on at 100 times when all the different cells see BMP, maybe one of the cell lines just expresses just a touch more of it. So these patterns can be used in combination in that way. So this is a transcriptional signature that is specific to the H9 ES cell line. And if we look at the advantage to H9 being a famous line is that many different groups around the world for years have been studying. So what we have here are 6 different studies, each of which studied multiple human lines, one of which was H9. So in 2013, these lines were studied at the NIH, again in Ron Mackay's group. H9 is also called WA9. Here, in a decade apart, the H9 line was clearly expressing the same signature as it does now. So this is more RNA level data. This is DNA methylation data. Again, H9 annotated as WA9. It has low methylation at the genes where it has high expression, sort of an inverse relationship we might expect here. So here we have already the level 1 of a mechanism that these transcriptional signatures that are specific to individual humans, and I say humans because we actually confirmed that the expression of these transcriptional signatures we can observe in a dish are actually observable in the mature tissue of the donors. So we have another experiment I won't show you, but we had at the Lieber Institute, we had cells in a dish, the iPS cells from people's whose brain, postmortem brain tissue we also had. So if we had a brain or any mature tissue from H9, we could do this. H9 is an ES cell line, so no human was ever made from these cells. But in iPS lines, we've showed that. So, and again, just to move on here, these are other studies where H9 was contrasted to other lines, and the transcriptional signature that we extracted from H9 is present in every lab that we've ever seen sequence RNA from H9. So these are like you said, there's something about the scenario you were talking about is looking at pluripotent stem cell lines and then looking at genes that are to be involved in the clinical features of the person who gave that line. So, and this is, I think, really where we all are at the very beginning here. We really don't know how to connect what is happening to person A at 50 years old in the clinic and connect that to whether it's transcription or epigenetics or anything in their iPS line or cells. So I think what you're mentioning is a super interesting finding that you can already see the disruption in pluripotency of genes to be relevant clinically. And similarly, our transcriptional signatures of individual lines and individual humans in pluripotency being relevant to their differentiation is another way of trying to say that we'll be able to understand from simply looking at pluripotency what we might know something later, whether it's all the way down in a clinical syndrome, pluripotent cells might do when we give them a particular path to go down. And so I really think getting the data for all these systems together, I don't think there's another way to start understanding this. And
a quick question: how do you actually isolate the signature? Because when you work with NEMO in this way, I mean, I understand putting a group of genes, differential gene expression, whatever, but how do you inquire? How do you query the data set for a specific line? It's almost like a pattern or something, right?
Yes. So yeah, I'll tell you kind of the flow of what happened here. So step 1 is get this data set into NEMO, which is just a matrix of all of your cells and samples by all of your genes. Step 2 is to upload the signatures that we developed offline. So offline meaning this same data set here, these 6 lines that we're studying, we ran an NMF on them. So that broke apart the gene expression into different patterns. Now, what does that give us? That gives us a second matrix where we have 22 patterns, and then the other dimension is genes. So this G and this G are the same. So now pattern 13, for instance, has a weight for every gene in the genome. And so we've uploaded the data. That's this frame here where the data is shown. We've uploaded the gene lists from our 22 patterns. That's here. And when we create the right web link, this picture is now showing the strength of these signatures. Is that kind of what you were asking, or more practically, how do you do it in the NEMO system?
Yes, thank you. I got the idea.
Okay.
You did it offline. You don't do it at the moment.
That's right. Currently, we don't have the ability to say, take this, get into pieces, and then project those pieces. That's definitely the next piece on our mind. So currently what you have to do is come here and you upload your gene expression data set. And then you go to this other piece. I know this is very small, but there's a button here to upload a gene expression data set. And then here, upload what we call a gene cart. So that's the weights of genes or the list of genes. And once you have both of those in the environment, you can combine any data set with any gene cart. So once you have hundreds of data sets and many signatures, the game gets very big and you can really ask any question you have of any data set. But yeah, eventually we really want to have this all contained here, where if you upload your data set, you can decompose it in Nemo and then project your decomposition from your own data set into related data sets. Yeah, dying to have that up. But in the next year, I think we'll be able to have that up here. And I think maybe we're closing down on 3 now. I'm really glad some of you were able to stay, and really enjoying this conversation, and hope for some more. Yeah.
Sorry, Carlo, I know it's late, but I had
No,
the
not at all.
question because I'm really curious on these results. Did you happen to upload any transcriptomic data set of naive pluripotent stem cells? Because I'm wondering if you have any evidence that this kind of transcriptional signature can be erased if there are protocols that should reprogram everything.
That's a super interesting question, and I have a little bit of an answer for you. Let me pull out here. So I can show you a little bit of, we don't have naive and primed, but we have something that is almost naive and primed. So this here is a very simple analysis. Let me just zoom in a little bit. Oops. Come on. Okay. Over. So this versus epiblast stem cells in the mouse. So it's not naive and primed as in a human naive and primed where you use 2i or 5i to induce these different states, but it is close to say it's almost the original naive versus primed. And what we see in the context of what we've been discussing, so if you do a PCA analysis of ES versus epi, which would be, say, naive-ish primed, a naive high versus primed low here, if you take this signal and project it into the 300 next-gen lines, what you see is what we might expect here. Let me get my pen back out. So it's highest back here and lowest down here. And what we've noticed in other dimensions of looking at this is this basically is the darkest cells here with the, so that would be the most epiblast stem cell like signature versus ES back here. These are higher in retinoic acid signaling. They are higher in sort of lineage forward places. So the cardiac we saw down here, the neural we saw down here. So this is sort of primed versus naive in this map is almost in this dimension here. And whether or not those, you know, I think maybe the question, and correct me if I'm wrong, you were wondering if those person, line, or donor-specific transcriptional signatures are still identifiable in, say, a naive state. My personal guess would be absolutely, and maybe even more clearly so, but we don't have a case in which we have defined the transcriptional identity specific to a donor and then taken those specific cells and pushed them through a naive protocol. There's plenty of naive versus primed data out there, but we don't not only need the naive versus primed in one particular cell line, we also need enough data and observations to get a transcriptional signature from that line. And I don't think we have that collection of data yet.
Thanks a lot.
Yes. Super interesting. Yeah. And you know, again, one other thing we've noticed, in fact, Silke Kim, who's on the call and the first author of this paper whose figures we're looking at, she recently made sure I completed a new experiment, which was to see if we could identify the transcriptional signatures of the individual lines if we used only the pluripotency data, not the differentiation data. And they actually get even, they get very strong. They don't project as well. When you look at them in other data sets, they're not quite as clear. But if we ask for 8 patterns in a set of 6 pluripotent stem cells in self-renewal only, 6 of those 8 patterns are simply the line-specific signatures. 6 of 8. So almost everything that is going on in pluripotency is about a cell line's personal identity. So that's why my guess would be that if you could get to a naive state with all these lines, by 5i, 2i, whatever you do, that I think it'd become even more clear.
Can I just reinforce
Please
the point we're discussing here? This is a great conversation. So, I mean, it looks like the way to think about pluripotency in this system, right? I mean, what happens in embryos is another question, but is that actually there are many different pluripotent states. There's no ground state, which is, I think, what we're discussing. And in terms of creating a kind of matrix where you can really look at variability, strikes me that might have some quite interesting consequences. But at any rate, for the point we're at now in this, talking about human variation, it seems like this is a really nice place for this conversation to end. The variation seems to be intrinsic to the pluripotent state.
Yes,
and isn't that right? Isn't that what we're just basically all discussing?
Yes.
I think, when I... No, I was just going to say, I mean, this is not news to a lot of people. A lot of people think this way already, but it seems like it's still worth emphasizing.
Yes, indeed.
That's right. That's what PC1 is about.
Yes, so here we've taken the PC1 from the next-gen data, so the PC1 across 300 pluripotent lines, and simply mapped it onto here the mouse and here the monkey developing embryo. So the low end is pluripotency in the rectoderm, and the high end is mesendoderm. In very clearly in the mouse and monkey, it's both cases. And again, this is simply natural variation in human pluripotent stem cell line. And it's mapping,
it's like the first most important decisions that everybody has to make. That's
right. And all these cells are about to make it, and they're wobbling, leaning toward this way or leaning toward that decision.
And has repercussions for all your life.
Yep. Yes. Who is it that used to say the most important time in your life is not birth or conception, but gastrulation?
Lewis Wolpert.
Lewis Wolpert.
Yeah. Fair's fair. But what we understand about gastrulation, though, right now, is that in the case of, if you think about all these images of the embryo, we're also sort of looking at a time map. And it's as if the pluripotent state, this relates to your question about naive and primed, the pluripotent state is kind of biased towards a cell which at this very far sort of left-hand corner of this purple pluripotent state in the Pijuan Sala, that's right. That's what you might call the ground state. And when you passage cells, how they go through this state seems to be a very important issue. And actually, if you just go down to the bottom of this figure here, I didn't expect to be talking about this figure this morning, but still, here we go. We have our own data on this, right? So because these are our own data on black is self-renewal and red is neurectoderm, 2, 4, 6 days of culture in this sort of cycle of culture we use. So that's right at the top here, that where you were talking about the naive state exists. And so that this all becomes very, very, very interesting. How they cascade out of this. To say one more thing, perhaps, all the work on lineages in the brain tell us, that's kind of why I was emphasizing this point about timing, is that the cells in the forebrain go through their use of eomesodermin, but they don't do it until for weeks, right? Until the neuroepithelium has reached that point. So there's a whole the brain is this kind of protected place. And when you showed the images of the mouse embryo at that very high resolution tool from the Beijing group that's just been developed, that's very interesting because there you see the relative size of the head and the body. And this image here where you're showing the heart, this is all very ancient. So all of this is really, I think, going to be much more deterministic than we say that these mammalian embryo, let's say C. elegans,
Sure.
we might find when we look carefully at lineage data at our hands, so alongside these data sets that we're discussing today, it could turn out that the cell biology here is much, much more deterministic than we know. Anyway, I'll just let that speculation hang out there. But this is very interesting. In Xenopus, at early times, a group in Europe predominantly, but with a lot of influence from Genelia farms, has looked earlier in gastrulation, where the heart cells are clearly a very, very major early component of the Xenopus embryo. So this, if you go slightly earlier, just up above Carlo, right? See there, the heart there's the head, and then there's the heart, and then there's the body. That's the way these things are designed. And so very early on, we should be able to see this heart signature. And actually, if we all had the appetite, we could go and look now and really explore in some depth what Carlo was saying. Where is this heart signature in coming up when we put the cells through this cycle of renewal?
In the pluripotent stem cells.
Yeah. It's very cool.
Yep.
Thank you.
And so I'll send some links to these resources in this follow-up to this. And I'll also include some of what we didn't look at all today are the neocortical. So this is obviously a little bit before any neocortex is going on, but we have similarly rich data sets in both in vivo and in vitro data on neocortical development. So I'm very, very excited that we're having these interesting conversations and to get people into these data environments and have them used, because the data all exist, but they're not easy to use right now. So trying to get it all together. Thank you guys so much for hanging around. Really enjoying these conversations, and see you in a month, if not before.
Thanks a lot.
guys.
It was great.
Grazie, Nicola. Ciao.